Hello professors, this is SaidAbdellatif and I would like to present methods and results of SDSPAGE experiment. Starting from the methods, the IMAC immobilized metal affinity chromatography is used to produce the crude lysate and elution samples. Starting with lysating E. coli cells which produce GFP by adding B-pair reagent and incubate and then centrifuge and separating the supernatant, and we take a sample from this supernatant and label it as crude lysate. Then this supernatant is mixed with NINTA slurry resin, and this resin would bind to GFP. So after that we centrifuge and wash 3 times consequently to purify the protein, and finally we elute the protein from the resin by adding the elution buffer, which contains high concentration of imidazole, which would replace the GFP from the resin. And now the supernatant contains the resin. After centrifugation we separate the supernatant and label it as elution sample. Elution sample and crude lysate sample would be stored on ice until we use them later on SDS. So SDS, we start with preparing the gel from monomer of acrylamide added to bis acrylamide in presence of catalyst, PMID and APS to prepare our acrylamide gel, which is prepared in 2 different concentrations: resolving gel, which has higher concentration than stocking gel. On the meantime, we prepare and assemble the electrophoresis cell, and then we pour the resolving gel, and after solidify we add the stocking gel, and in the meantime we put the comb to leave spaces for the wells. Then we should have prepared the samples by adding 6 times SDS loading dye to each sample and mix them well and heat to 95 degrees Celsius, and then we put them on ice until loading. Loading wells would be efficient and quick as possible. Then we run the electrophoresis on 200 volts for 30 minutes. After finishing, we disassemble the electrophoresis cell safely, and then we wash the gel with distilled water to remove SDS, and we transfer the gel in gel box to the ChemiDoc imagery to take UV pictures, and these photos would be viewed using the lab image software from BioRad. And now moving to the results of SDSPAGE. Starting from lane number 1 and 2, this refers to the ladder, precision plus molecular weight standard ladder from BioRad company. And then number 3 is our BioRad standard protein, which contains 3 known molecular weight proteins. A is bovine serum albumin with around 66 kD, and B is egg albumin around 35 to 36 kD, and C myoglobin with around 17 kD. And now moving to number 4, which refers to the crude lysate. It has very pale but many bands, which refers to the GFP protein, the much of proteins coming from lysating the cell, the E. coli cells, and this is why there are many bands appearing in 4. And then number 5, we have several bands instead of one pure band of GFP protein, and this may happen due to dividing of the protein to subunits. So each subunit would appear as a band. Then moving to number 6, which is referring to ion exchange protein. We have good purified protein with band appearing at the same molecular weight of myoglobin. And finally, number 7, gel filtration protein. The protein doesn't appear with any bands, and this may occur due to poor mixing with the dye before adding it to the wells, loading the wells. Finally, my recommendation is to mix properly the sample with the 6 times dye, SDS loading dye. And when loading the standard known protein sample, it should be in less concentration than what was used in this procedure to avoid overloading in the wells which appeared in lane number 3. Thank you.